So, thank you so much for joining us, Darshit. Welcome to the seventh episode of Proactive Aging, where we discuss topics around health, wellness, senior care, and much more. The intent of the podcast is to, provide nuggets of wisdom that people can incorporate in their own lives. At Saya, we run structured health programs to help people pushing 50 or above that
Mhm.
improve their baseline health and ensure that they avoid, manage, or even try to reverse their chronic diseases and conditions with a
Mhm.
special focus on diabetes management and reversal. So, thank you so much for joining us, and I'll get right into it. I think the topics that we'll definitely be covering will mostly center around gut health.
Sure.
So, thank you so much for joining us.
No, of course, Karan. It's my pleasure.
So, I'll get right into it. What was the inspiration behind creating the Decode Biome? Where did you see it first, and why did you feel the need to bring it to India?
Right. No, absolutely. The story is a little bit long-winded, right? In very early ages, and I think about my mid-teens, I got interested in biology, particularly so with cancer. That's what led me into biotech in general, right? And just after my undergrad, I got really excited about computational biology because that was coming up as the next frontier. And back then, the biggest story in this space was DeepMind, which is a Google company for protein folding. So that essentially got me into computational drug design. And I was not in the country. Then COVID happened. I came back to India. And in fact, COVID was the biggest blessing in disguise for me because for the first couple of lockdowns, being a computational biologist, I got on to work with a university in India on a project which was on metagenomics. So there, what we were trying to do is we were trying to look at all the freshwater bodies. So Pune was the city. Pune has multiple dams, lakes, rivers, right? So we were doing the bacterial diversity throughout these freshwater bodies, trying to figure out if there are certain antimicrobial genes which are actually entering the food chain through water systems, right? Because AMR is something that is being talked about a lot these days, considering the unjudicious use of antibiotics. So that's what really got me excited. For the first time, I stopped looking at this world in terms of organisms and in terms of ecosystems, right? And of course, that got me interested. I started looking at where all microbiome really exists, right? And there was soil, there was marine, there was fresh water, and then within the human body, there was gut, lungs. Many people have tried to report blood, but I'm not completely in agreement with that. But you will see vaginal microbiome, oral microbiome, then cerebrospinal fluid microbiome. So it was really a big exposure to a completely new field where I got to do it with a lot of practical experiences as well. But coming back, like I always wanted to work on the human biology, human diseases, right? And particularly aging, because that's what most of my curriculum was as well in one of my postgrads. So that led on to an understanding that, okay, two, three things could be put into the same bucket, especially in India, where biotech data generally is at dearth, right? We do not get enough data banks where we can work with. So that was my first understanding. Now, this is 2020, and hallmarks of aging were the topic, are still a topic, right, in the scientific community. But back in 2020, microbiome was not a part of hallmarks of aging. At least that's what the scientific community decided, that we will not be going forward with that. But my instincts kicked in, I would say gut instincts at this point, and thought that if microbiome is such a vital player in all the other environments, surely it should be important in the human host metabolism as well. And there was enough evidence there. So I started looking at microbiome purely from the perspective of aging before it was considered a hallmark of aging. And then boom, 2022, we get that as a hallmark of aging. So my gut instincts were actually right. And we would see that probably it's something to do with short chain fatty acids later on in the podcast. But yeah, that's really how we all framed up. about looking at microbiome and aging very specifically while building in the same space as well. So our R&D about that started in about 2021, and I would say we are only still 1% done.
Wow. You were mentioning this, the hallmarks of aging, right? Can you elaborate on that more?
Surely. I think over decades of research, right, scientific community overall throughout the world decided that there are some mechanisms which are central to human aging, or something that you can depend on when you're looking for evidence for aging and longevity in general, right? So that framed up the hallmarks of aging. Now, back in the year around 2000, 2002, there was a paper called Hallmarks of Cancer, right? And it really propelled the field forward because now people had a consensus on what I should be looking for when I'm treating cancer or when I'm researching cancer. So that breakthrough moment for aging and longevity came in 2011, which was when the first hallmarks were out there. So we all keep hearing about things like telomeres, right? Or genomic instability, or chronic inflammation, or loss of proteases, which is making the body's capacity to make protein molecules, right? So these are all now a hallmark of aging, which is people study these mechanisms when they are doing studies around aging and longevity. So 2011 was the first time we had 9 hallmarks of aging: mitochondrial dysfunction, telomere attrition, genomic instability, chronic inflammation, loss of proteases, epigenetic alterations, right? So these are altered cell communication. So these were the 9 that were jotted down back then. In 2022, we had a revision of that. And now there are 14 hallmarks of aging, and some of the new ones that were added, and one of them was actually microbiome. So it's a very interesting thing that happened. They used one particular study in evidence. So they took some mice, right? And they were so there was a group of old mice and young mice, right? When they took the stool of old mice and implanted it in young mice started aging. And when they did vice versa, certain signs of aging or hallmarks of aging reversed in the old mice as well. So that was the first definite proof that microbiome has a role to play in aging. And there was mechanistic evidence for that through my studies, and that's why it got added in the hallmarks of aging.
And is this hallmarks of aging, is this maintained by a scientific body, official scientific body, or is it just a consensus, uh, you know, document or consensus book of guidelines that is maintained by, say, a university or a bunch of them? I don't know.
Yeah, so actually groups of scientists from multiple universities come together to form a consensus, and they usually publish it. So hallmarks of aging, you will see 2 papers published by the heading Hallmarks of Aging, one in 2011 and one in 2023. So that's where, and lots of scientific leaders who are working on aging and longevity come together to write this single paper. So you'd see about 40 odd authors to a single paper, which says that everyone is aligned that this is what we consider as signs of aging or biomarkers of aging, if I have to put it like that.
Understood. Understood. And so, what makes first of all, can you describe the process of the gut microbiome test and why it's different from, say, a regular stool test or even, say, something like an H. pylori test that we typically get in India? I'm sure even the H. pylori is not as well known, but why is it different from a regular stool test to begin with?
Right, right. So I think we need to look at certain things when we are talking about gut, right? Now, gut is a very elongated organ. It starts from your mouth right until your anus, right? Maximum of the research kind of happens in the small and the large intestine, because that's like a metabolic factory. That's where everything in your body is going to start with, right? So small and large intestine are the main places. Now, there are different things you test when you're looking at a gut, right? If you would see that occult blood test is something very common, which we do, and it's very cheap and available at a very mass level, right? That essentially checks for blood in the stool, right? Which tells you about a few markers of inflammation and other things, right? So that's actually happening on how much human blood is secreted into the gut, right? So that's one aspect of gut testing. The second aspect of testing is looking for certain bacteria. You mentioned H. pylori, right? H. pylori is a severe gut infection, right? So when you're looking for an H. pylori test, you're looking for an H. pylori, nothing else, right? You're very concentrated that I want a diagnosis for H. pylori, right? Now there are third kind of tests, right, where they look for things like zonulin, calpopactin, beta glucuronidase, right? So zonulin is basically a protein which is made by your body to make sure that the linings of the organs are tight, right? So that they don't loosen up and something doesn't escape out of it. So that's for zonulin. Similarly, there's calpopactin and glucuronidase having their own understanding of those markers. So that's the third kind of test. Microbiome is something very recent, right? So there are two ways to test microbes presence in a certain sample. Could be soil or could be stool, right? One is culture-based methods, right? Every time we think of microbiology, people put up things like, those Petri plates, the glass plates and everything. Right? And there are certain bacteria growing onto it. These are called culture-based tests. When you're looking for E. coli, when you're looking for H. pylori, right, or a certain bacteria, you could use that test and it will give you a definitive answer. But about 99.9 organisms out of 100 that we see in the world, which are bacteria, cannot be cultured in a lab at all, right? That means that you'll never be able to catch them on a Petri plate. For that, the same technologies are used, which we use in human genome sequencing. So lots of us are getting gene testing done these days, right? That I have this panel of genes. It tells me predisposition to a certain disease and all. So similarly, same technology when applied on your microbes, which are living in a certain environment, is called microbiome analysis, right? And there are two ways of looking at that as well. There's microbiota and then there's microbiome, right? So microbiota are the species present. Microbiome is the overall genetic content that is present in those bacteria. So this is where microbiome testing comes, right? It might not be able to, it will definitely predict certain bacteria like H. pylori and all, but lots of others as well. And now we are understanding that it's not often one or two bacteria, but lots of other plays a role. And the way they maintain internal ecosystem as well affects our health. So microbiome is more about testing the overall presence of different bacteria, fungi, archaea, viruses in your gut, looking at how they interact with each other, how they interact with the food that you eat, and what do they make after interaction, and how these metabolites or byproducts eventually affect your health. So that's microbiome. H. pylori or microbe testing is a very specific diagnosis for a certain bacterial presence. And there are other markers which are related to your intestinal health, which are not essentially microbe dependent.
Got it. So when you speak about good and bad bacteria in the gut, right? Talk to me if I'm as if I'm a third grader, for instance. So if as a third grader, what role do you think is played by good bacteria? What role do you think is played by bad bacteria? And how do they interact in the gut, especially when it comes to what we consume and what the food that we put inside our bodies?
Right. Okay. So the general understanding of good and bad bacteria comes with two kinds of species that reside in your gut, often called as probiotics, again a very common word, and pathogens, again a very common word, right? So these is a broad classification. Now, if we have to say why do we call probiotics probiotics and why do we call pathogens as bad bacteria, is because they release certain substances, right, which either affect you negatively or positively, right? For example, probiotics or the good bacteria are more known to release metabolites or byproducts which are beneficial for gut health, right? Could be a butyrate, could be an acetate, could be a propionate. These are the three words which will very regularly hear throughout the podcast, right? Because they have the maximum evidence of they helping human host positively. That means it will help you with the inflammation. It will help you with your brain health. It will also help you maintain lower inflammation in your body and stuff like that. But pathogens, on the other hand, would release something more toxic in nature, something like a lipopolysaccharide or LPS, right? Or other inflammatory molecules which are going to cause problems in your gut lining. It's going to elicit a lot of immune responses, right? So those are the issues that it can cause, and which is why they are categorized as bad bacteria, right? But we tend to forget a third class of bacteria which no one talks about because no one understands them, right? The good and the bad bacteria. Let's say you have 1,000 species in your gut, right? 10 of them are good and 10 of them are bad. What about the other 990? Right? They are also part of your metabolism. These are called commensals, or someone who is part and parcel of your body, and they help in good. If your overall environment in the gut is good, it will help you. If the overall environment is bad in the gut, then it will not help you, right? So it's stuff like that. So that's the third category of microbes we tend to forget, and that's where maximum insights are.
Got it. So if I have to give an analogy, it's the commensers are basically the public that is misled, and the pathogens and the probiotics are the ruling parties that are making the decisions.
Absolutely. I mean, we tend to remember the fame, but not all the other guys who are making the machinery happen, right?
Got it.
Yeah.
Okay. That's nice. Yeah. And what is the difference between a gut microbiome test and a food allergy test? While I know now, having done both of those, what the difference is, a lot of people who I speak to are not able to differentiate between those, right from even the method of collection to actually what the findings are. So, can you explain the differences between both? What unique purposes both serve, and why I say either both? What do you think if one had to get one of those two done? What do you think is superior?
Right. So, really depends on your symptoms, to be honest, right? We need to understand microbiome is all the microbes in your gut, and then there is the human host where they live, right? Now, when you eat certain kind of food, there are two types of responses you can expect. One is your body reacting to it, and one is the microbes reacting to it, right? When we talk about the food allergy test, and lots of people come to us saying that, can you test food allergy with this test? And we often say no, because you can't. So what happens with food allergy test is, you eat a certain food, the food has certain things which your immune system doesn't like, so it reacts by either giving histamine, right, or related metabolic outputs of allergy, or something like that, right? So that is basically your body reacting to the food, not microbes essentially, right? So you will see that often for allergy testing, they'll take your blood, right? They'll look at IgE levels, which is again one of the molecules that your body secretes when it is having an allergy, right? So it will look at IgE, it will look at histamines and stuff like that. So that's your body reacting to the food. That is allergy. Microbes is something else altogether, right? When it comes to microbiota, it's more about when a certain bacteria consumes a certain food, it releases something, right, as a byproduct. Okay, so that's what we are looking at essentially in the microbiome test. So things like bloating, right? When we experience things like bloating, or things like flatulence or gas in our stomach, right? Those kind of symptoms are more about the microbes reacting to a food, right? But when you start getting skin rashes, sneezes, right, irritation on the skin or anywhere, right, those kind of symptoms are usually more about allergy than about microbiota. Is your body's response to food rather than microbes' response to food. So that's how, at least I have understood the two tests, and that's how we suggest people which test they should take and why they won't get an answer from a microbiome test.
Understood. Understood. So they serve very different functions from what I gauge.
Absolutely, very, very different things. Yeah.
And now again, I'm stepping into the shoes of my third grade self, and I'm going to ask you another very. While we all have been hearing that the gut is the second brain,
Hmm.
to a third grader, how would you explain why consuming the right foods for your gut impacts your brain health?
Right. No, definitely. I mean, to a third grader, my explanation will be a very simple one, right? When you eat good foods, your microbes make good metabolites, or you would say good byproducts, which helps multiple systems in your body, right? So that's the simplest explanation. But how does it really connect to the brain? The answer might be a little bit more technical there, right? When and it's often related to the gut and brain connection is very evident in our everyday lives, right? People have cravings certain times, right? Those cravings are actually a crosstalk between your brain and gut, right? At the same time, you will see that when you start adopting a healthy lifestyle, those cravings go away, right? So your brain demands food that your bacteria is asking for. And if you have more good bacteria, then you'll be asking for more good food and stuff like that, right? So that's a very basic explanation. But lots of systems are interconnected when it comes to our body, right? Now, why is it called the second brain? Our brain is made of neurons, right? Our gut should be made of epithelial cells, right? But apparently, our gut has more than 100 million neurons, right? Which are actually brain cells, right? Which are more than what you have in spinal cord. Something we called enteric nervous system in body's physiology. So enteric nervous system is all these neurons lining up in your gut, right? And they're connected to your brain through something we called vagus nerve, okay, or vagus nerve. So there's a nerve that connects your gut centric system to your brain's overall multiple centers in the brains, right? So it could be basal ganglia, could be hypothalamus, multiple, right? So this is the main connection that forms it, right? Now, yeah, please.
No, no, please go ahead. Yeah.
Right. So one is that, one is the connection, literal connection between the gut and brain, which is through a vagus nerve, right? And we saw that there are so many neurons in your gut as well, right? They're essentially not helping you think, but they create a lot of ripple effects, which affects your brain, and that gets you to think better, behave better, have a better mood, or less fatigue and other things. How exactly also, I'll try to tell you, right? So that's the first place. The second is the inflammatory system. Inflammation, lots of common words again regarding inflammation these days, right? So that's essentially connected to your immune system. When your immune system reacts to something negative, it starts releasing cytokines or inflammatory molecules. So that's the second system. We have the vagus nerve, we have the immune system. Again, the maximum concentration of immune cells in your body are in your gut lining.
Okay.
So that's the second system we have. The third is the gut lining itself. When they break, certain things exit and they reach to places where they shouldn't have reached, first of all, right? So that's the barrier integrity. Everyone talks about, oh, leaky gut is fake and everything. But I do really believe that gut permeability plays a major role, right? So that's the third aspect of it. Now, let's say when you eat something nice, which your body likes, a fiber, okay? Fibers are the best microbiome food ever made, or fermented food, right? So what happens is these fibers are broken down by the microbes, right? And then they will release butyrate, acetate, and propionate, right? These will actually help you maintain your blood-brain barrier integrity so that they don't leak. And if blood-brain barrier leaks, then things from your blood start leaking into brain, causing inflammation in brain, okay, or certain kind of toxicity in the brain, if we have to keep it very simple. So that's the first way how it affects, right? The second way how it affects is that, let's say you eat something bad, okay, a processed food or something. Your microbes are going to digest it and produce toxins like LPS or IL-6 or TNF alpha. These are all inflammatory molecules which will exit your gut. It will enter the bloodstream and it will reach places. When these things reach the brain again, right, they cause a lot of neurotoxic effects into your neural health or the brain cells again. And the third thing is the barrier loss and the gut integrity loss. If that is lost, again, things will escape into the blood, reach the brain, and cause a problem there.
Got it.
But our understanding, or most of the things that you'll hear on influencer content, is that your gut produces serotonin and dopamine, which reaches your brain and it helps you. It's not exactly like that, right? What's produced in the gut stays in the gut. It causes certain downstream effects, which ripples out through a series of metabolic pathways and cause a problem. If you ask me, acetate, butyrate, and propionate are more related to the gut brain axis than the serotonin and dopamine itself, right? So there is a certain kind of misinterpretation we do because we hear serotonin and dopamine in context of brain, but actually they are very much helpful in the gut motility and how it interacts with the vagus nerve, so things like that.
Yeah, I'm guilty of that. I always believed until now that your gut is directly responsible for releasing these hormones. I mean, of course, I think one of the reasons that you feel this way is your gut. But now that you explained to me, the series of processes that
Yeah.
actually go on connecting the brain and the gut, I think now it becomes very, very clear. So for those people who are saying that, all of our dopamine, our serotonin, a lot of which is linked to the gut, uh, if that is not a direct effect, where does that come in?
Right. So serotonin, and it is a sure shot fact that a lot of serotonin and dopamine is actually produced by your gut itself, right? There are neuron cells, as we talked about in the gut, right, which are producing it. Or when you take amino acids or proteins which are rich in tryptophan, right, that will also convert into certain kind of precursors of serotonin and dopamine. They are produced in the gut, and they help with things like gut motility mainly, and gut permeability, and the immune system that is interacting with the microbes and their metabolites in the gut lining. That's where they play a lot of role. Even so, vagus nerve has one end in the brain and the other end in the gut. So the gut serotonin and dopamine is going to play a role here, which cascades into different metabolic reactions and reach the brain and cause inflammation there. So that's the pathway it does. I'm not saying the gut derived serotonin dopamine doesn't affect brain health, but it does, but in an indirect way rather than a direct. You don't see serotonin molecules traveling right from your gut up the vagus nerve, entering the brain and causing you sleep. That's not going to happen.
Got it. So, there was also this entire study that someone like Bryan Johnson did, right? He's been doing multiple practices and running multiple experiments and testing the data on himself, understanding if if this is working for him or it isn't.
Hmm.
One of the things that he did was he's doing something called the hyperbaric oxygen therapy. I think you enter this coffin-like chamber and it's set at a certain temperature and
Hmm.
stays there for 90 minutes.
Right.
And over time, when he did that, he found that the balance of good gut microbiome, keeping all else constant before and then after doing that,
Hmm.
changed. What could be the direct impact between such a practice, and are there any such practices apart from the HBOT that have shown positive results on tilting one's gut microbiome, gut bacteria towards having more positive bacteria in their gut?
Hmm. No, I mean, try to look at literature like that, right? And honestly, there's not much direct evidence, to be very honest, right? Somewhere where you'll find a strong evidence is that HBOT has helped with people with ulcerative colitis. So we talk about IBS a lot these days, right? So when IBS flares up, it causes something called ulcerative colitis, which is basically ulcers in your colon, if I put it very simply, right? In those cases, HBOT has helped, right? But for generally someone who is healthy, right, how HBOT would modulate microbiome, we are still unclear of, and we don't know enough about. Obviously, Bryan Johnson might have seen that, but he does a tons of other things, right? And it's very difficult in one person to point down that this happened because of this, right? And that causality in gut microbiome itself is a big bane of our existence. Everyone who's in the microbiome industry, right? On the other hand, a couple of my study, in fact, show very opposite results, right? That HBOT would cause damage. It increases mice's susceptibility to something called C. difficile, which is like a lethal disease or infection, right? But that's still in mice, and mice microbiomes work a little bit different from that of human microbiome. So we're not very clear of. But that said, I think HBOT for health practice is something we don't have evidence for. HBOT for a very specific condition like ulcerative colitis, we have initial signal that it can help.
And say HBOT might be a more extreme practice, probably unaffordable for most of us Indians as well. But even say daily practices like sauna, ice bath, cryotherapy, which is again very unaffordable, and there are centers doing that. Do they have any of them shown to have any positive effect on ensuring that the balance of good bacteria increases in your gut and the balance of pathogens reduced in the gut, or something around those lines?
So I'll give you twofold answer to this. One is the direct understanding what we have about different biohacking techniques and what it does to microbiota. Secondly, if I really can trust these results or not. So these are the two ways I'll answer here. The first one is uh, things like sauna, right? So sauna usually would dehydrate your body, right? And that would cause negative effects on your microbiome, right? So if you are sauna plus hydration, maybe it works, maybe it doesn't. Very low evidence again for that. People are saying that photobiomodulation, which is lights, right, are affecting how the gut microbiome is shaping up. But again, we don't have a lot of clean evidence and answer for that as well, right? Then we talk about cold therapy. Now, cold actually subsides a very important bacterial growth, which is Akkermansia mucinifila, right? It has a big role to play when it comes to handling glucose in your gut, right? So it is actually subsided when you take a cold exposure. So again, there could be certain kind of negative effects, but I don't say that these will destroy your microbiota. We understand it very little. Short term might help, okay, and if it's paired with the right thing, then it will help. That said, the maximum gut microbiome change which you can achieve is by food or hygiene, because that's your maximum exposure to gut microbiota. So now, if someone is doing a study with something like a HBOT or photo modulation on a group of people, and let's say their bacteria shifted, right? But we do not understand what they had on a daily basis in terms of food or what kind of environment they were exposed to, it will be very difficult to say that this caused this. So that's the problem we are facing with general things. That said, if I have to suggest one biohack for everyone to improve their gut microbiome health, eat fermented foods.
So
That's
South Indian food like idli, dosa, curd, obviously, I think is uh,
yes, yes. But something that is fermented for a longer time would usually help, right? So things like, I mean, kombucha is one trend. Sauerkrauts, or we have kimchis, we have pickles, right? These things kind of help with the gut microbiome health. And of course, a lot of strong fibers. So basically, your veggies and a little bit in a raw form, right? Which you cannot digest yourself, only microbes can, and that will help you release things like acetate propionate butyrate.
got it. So I'm going to open. I got the Decode Biome test done, and I'm very happy to open up my information to everyone here. And I'm going to open up my report, and I want a little help in, taking my report as the guinea pig of sorts to help everyone else watching uh, interpret what, say, the gut microbiome test interprets and how it applies to me as per my report.
Hmm.
And say any two, three top level recommendations that you can make for me as well. So let me know when you can see my screen. So obviously this is my report, and I'm happy to share my findings with the with the audience that is watching.
Hmm.
And I want you to, as it says, as you say, decode the report to me and decode the report and help me understand what you know what these different
Scores.
scores indicate. Now, when I first saw green, like any other person, I was happy and I was satisfied. I was like, okay, maybe I'm definitely not, there's nothing wrong with me. But that said, what would a red disease susceptibility score or a red gut microbiome diversity score indicate?
Right. All right. So DSS, first of all, disease susceptibility score, right, is something we developed as a ratio, right? So ratio of harmful bacteria to helpful bacteria, right? So that's what we are essentially looking at. So taking certain disease cohorts and what bacteria are most occurrent in those disease cohorts versus people who are protected from disease and what kind of bacteria are overly represented in them. So usually good to bad का ratio है, ठीक है, about disease cohorts, and the more you are on the positive side, so the scale starts from 0, green is all positive, and negative is all the red one. When you have a negative score, essentially the bacteria that are promoting the disease versus protecting you from it are much higher. On the other hand, the green has more bacteria which are protecting you than harming you, right? So obviously good. And to be very honest, 3.22 on the positive scale is an extremely good score, right? Usually would see it in the point of 0.5 to 1.5. That's the average we see generally in a healthy person. But 3.22 is absolutely great, right? And that shows that you have maintained your gut good enough, at least so far, right? Consciously or unconsciously. Uh, yeah. And so diversity score or Shannon index is used globally as a marker to understand your diversity, how many different kinds of bacteria are living in your gut, right? Uh, Diversity for the longest period of time until I would say 2017-18 was the golden marker to say if your microbiome is good or bad. I mean, to be honest, I would not say healthy, but good or bad, because healthy as a definition is still very vague in microbiome, but good or bad. Now, higher diversity is always good because just imagine this, right? Both of us run startups, right? In the early days, we have 5 people with 5 different skill sets, so we can get 5 things done. But when we raise the team to 15, 20, 25, and we have multiple skill sets, more things get done on an everyday basis. It's the same logic. The more different kinds of bacteria you have, the more spectrum of metabolism you can perform, right? So the higher diversity is always very good. Again, 3.77 is a rare sight in India, particularly an Indian living in a tier 1 city where already there's a lot of microbe loss, right? So an urban gut is one of the most susceptible gut to different conditions. And to have this kind of diversity score in an urban center means that you're doing a good job, again, consciously or unconsciously, right? Now, if we come to the next page, right, which is essentially the most abundant species in your gut, right? It's naturally my logic to report this. You'll not see very frequently in all gut microbiome reports, but my logic to report it is that if a population is dominated by a certain kind of bacteria, it's ought to be the most influential one for your health, right? So that's the basic logic. It's as good as if a country has a majority of a certain religion, there will always be a certain kind of biasness towards that religion, right? In that country. Very much like that, very vague example. To talk about religion is very tricky these days, but that's the example I would give, right? So which is why we report most abundant species. You'll see red or green tag right in next to the number that is given for that bacteria, and you will see a initial category which is pathogen, commensal, or probiotic, which we have already talked about previously. Pathogen bad guys, probiotic good guys, commensal guys who people don't care about, but they're actually very important players in your gut, right? And you'll see most of the guts are dominated by commensals. So if we come down to this, the red and green tag are for anti-inflammatory and inflammatory nature of the bacteria, right? Which is innately either inflammatory or anti-inflammatory bacteria. You'll see that first guy is Prevotella copri, right? At 23%. It is pro-inflammatory in nature because of the certain processes it does. But we don't get worried about it when an Indian takes this test and we report this as the top. Because you would see that in 90% of the species, because Indian guts are dominated by Prevotella, which actually helps with fiber diets. Ancestrally, we eat higher fibers in our diet and carbohydrates in diet. So our guts have been shaped to form Prevotella as a dominant organism for us, right? And which is clearly visible. Now, watching this guy out here is always a pleasurable moment for any gut, right? So Prosinid-Z is one of those important members which gets hardly talked about in the content that we listen to on an everyday basis about gut health, but it's actually a very important guy in maintaining gut homeostasis or natural conditions or environment in the gut, right? And that having them at 5% is also really a good sign, right? Now, again, something we don't see very commonly in Indian guts is having a probiotic organism dominating the gut, right? Now, for a gut that harbors about 700 to 1000 species, having 1% of a certain probiotic is always good. That to adolescents, which has a very good evidence that it helps you maintaining your gut health overall, right? So that's how I would look at it. Your overall burden for anti-inflammatory bacteria is also less. We need to understand any bacteria that digests carbohydrates will have certain kind of inflammatory effect, but that could be managed if you have the right balance in your gut, which you already have, right? So usually interpreting these gut reports are very convoluted and tricky because you need
Yeah,
to find where the right balance is as well.
yeah. So who better than the person who created it? And
It is tricky. I mean, the day the microbiome field succeeds is when this 20-page report becomes one-page report with a sure shot answer. That's the goal we are actually trying to achieve as well, right? So the next ones are the pathogens, the bad guys, right? And we don't see very high load of them out here in your report to be very sure, right? So if you'll see, all of them are very minutely present, right? And all of them are something we call opportunistic pathogens, right? Opportunistic and true pathogens. True pathogens means guys like C. difficile, H. pylori. Okay, these guys will short cause an infection, right? Opportunistic are the ones which are sitting on the side. As soon as you destroy your gut health, these guys are going to take over and cause a lot of inflammation. So, which is why they're called opportunistic. If generally your gut health is nice, which we have already seen for Upper, then you don't have to worry about opportunistic pathogens. They're going to just stay there, do the job as they have to. This is something we see again missing in the gut reports, right? Which are not detected. Pathogens which are not detected in your gut is good for you. And these are actually very much, what do you say, highly inflammatory bacteria or true pathogens, which can really cause a big time. For example, Campylobacter species would cause big time diarrhea, right? Clostridium is going to cause a lot of issues in your gut in terms of inflammation, aches, and stuff like that. Shigella flexneri, again, very much correlated with uh, diarrhea, right? So these guys are actually very bad for you, and it's not present in your gut. So you could cheer up a little bit there.
thank God, yeah, I'm less worried now.
Yeah,
I wanted to know a little more about the vitamins and the macros that that they
sure.
show. I think that's something common folk like us will be very interested in. I think if you go down,
Of course. Yeah.
it's
So right. Now, coming down to macro delegation. Now, the first section of the report is more about which species exist, which doesn't, what does it do, right? But most of the times, the analogy I give is that just because Darshit is good at microbiome science doesn't mean he's going to be good at all points of time at each aspect of microbiome science, right? So
right.
that's where the functionality comes into the picture. Sometimes we see a species, okay, for example, Prevotella in you, and we know that it's a great carbohydrate or fiber digester, right? But what if it just doesn't have enough genes to do its job, right? And this is the maximum contradictions we see out here in India. These people are dominated in Prevotella still have difficulty digesting carbohydrates, right? And particularly the complex ones, right?
Yeah.
So since the carbohydrate digestion is low, right, what you would see, and that's also because we do not eat a lot of raw food in our everyday lives, right? What you'll see is, experiment with yourself, take a bowl of raw veggies, okay,
Okay.
mildly boiled or anything, and consume it. You will definitely see issues in your gut regarding after having that flatulence, okay, or incomplete evacuation. Sometimes you will not be able to evacuate your bowel as much as you would like. And that is only if you go for pure raw food, right? Because the fiber digestion won't happen efficiently because there are not enough genes for that. So there'll be a lot of undigested carbs even after microbial fermentation, and that is going to cause a problem for you in terms of releasing certain gases, right?
I actually do notice that when I have these raw salads, kitchen garden and all these other raw salads that we eat with lettuce and sun-dried tomatoes and whatnot, and I've stopped eating those completely. I mean, everyone I know, everyone I know, they all eating these salads, and but I think it just didn't suit me. Each time I used to eat it, I used to feel a little irritation. You're right. I think there is some correlation there.
Yeah, no, absolutely. So even I had very similar issues, right? When we have raw salads, it will not get easily digested for us because the carbohydrate digestion potential of the microbe is very low, right? So how our understanding comes about, right? When we think of Prevotella copri, okay, lots of big scientific groups in USA studied Prevotella copri. They made their genomes and uploaded it on database, right? What we sitting in India do is we take your sample, we sequence them, compare them to that.
Yeah.
Now you are a Homo sapien. An
Yeah.
American living in Texas is a Homo sapien, but you all are different.
Yes.
Similarly, a bacteria that is found in USA versus the same bacteria found in India is genetically different. And if you'll see, a lot of our research, which we don't talk about because it's way too technical, happens at this level to understand what does Indian Prevotella copri really look like. Does it have the same genes as we know them to have, right? So that's the problem. Maybe in India, as we discover further and as we sequence more microbiomes, we'll realize that okay, Prevotella copri नहीं कोई और ही species कर रहा है और वो species मिल ही नहीं रहा है इंडिया में, ठीक है?
Got it.
अब देखोगे जो रूरल एरियाज हैं इंडिया के कोस्टल लाइंस पे, ठीक है,
Okay.
उनके माइक्रोब्स इतने ज्यादा डाइवर्स है और उनको ज्यादा इश्यू नहीं होता है सैकेरोलाइटिक ब्रेकडाउन में, विच इज अ फाइबर ब्रेकडाउन, राइट? एज यू कीप कमिंग इन रोड्स, और दैट्स द मल्टीपल पॉपुलेशंस दैट हैव स्टडीड, as you keep coming inside and towards tier 1 cities, suddenly microbial diversity is lost, suddenly the functional potential is lost, and that's why people are not able to digest a lot of food.
So there can be, I'm guessing there can be a lot of other determinants as well, because now while I live alongside the coast, I live in Mumbai, and and I don't know which one is it. Is it more on the urban side or on the
No,
coastal
no, rural. I mentioned rural
side?
towns,
Yeah.
rural areas of the this thing, or populations that are studying in Ladakh
Okay.
or Mizoram and all those areas which are kind of secluded,
Yeah.
maintained over a period of decades and everything, right? The biggest example, or my favorite study that I've read in microbiome throughout the world, is one that of Hasda tribe. So Hasda tribe from Africa is one of the most conserved tribes of the world, right? And they have not exposed themselves much to the world. They still live in the same environments they have been doing for past 3, 4, 5 centuries.
Mhm.
Their microbial diversity, Shannon index we saw, right, which is the diversity score
Yeah.
which you have at 3.77. They exceed 4.5, which is unseen in the
Wow.
urban populations. It's absolutely unseen in the urban populations, right? Plus, India ancestrally was meant to consume more complex carbohydrates, but we have moved to simple carbohydrates in the urban centers. When I say simple carbohydrates, it's things like bread, rotis, or rice, right? Which has become more dominant compared to veggies and stuff. Even when we eat veggies, I mean, how many times have you seen bhindi cooked at your place, which is literally fried to the level where there is nothing existing in it?
Yeah. Yeah.
Yeah. So we're not taking enough complex carbohydrates in urban centers, which is why it's not giving us much later on as well.
So when it comes to, say, protein digestion, now again, if I have to look at it as someone who's consciously tried to increase my protein intake over time, I'm vegetarian. I come from a Jain household, but through protein supplements and my food, I've at least tried to build a as close to a scientific blueprint for myself, at least when I'm having a in my daily meal consumption. Of course, weekends and other things when I'm eating out, then all of that goes out of the window. But on a day-to-day basis, when I'm living my normal life, I'm trying to get about 1.5 to 2 grams of protein per kilogram of my body weight. That said, now when I saw that my protein digestion is low, does that indicate that I need to consume more protein, or does it indicate that I have to consume better protein?
Yeah, exactly. See, I don't have a definition for a better protein. And honestly, if someone else is giving it to you, maybe they are not very well versed with how it happens, right?
Yeah.
So you will always see this contradiction when it comes to gut microbiome, right? We'll come to one page upper, right? Something we call enterotype. Okay. Enterotype is the type of gut you have. bacteroides, okay, Prevotella, and Ruminococcus, right? So Ruminococcus shows a more mixed diet, okay. If you'll go to Italy, Okinawa, or places like that, where the balance is very good between the food and that, you'll see more Ruminococcus dominance. In Western populations, which are more meat dependent, are going to have more bacteroides. Interestingly, we have seen that expats living in India have bacteroides dominance, which is very different. But most of the Indians are Prevotella dominant, right? That's what your gut can harbor. So you will always be digesting fiber better than protein.
Okay.
That said, if you want to shift your gut towards a more protein digesting environment, the protein powders are not going to help you.
Oh,
Okay. Because you need whole animal sources of food, which as vegetarians, I don't know if you are, but I am, we cannot take, right? So when you eat organ meats and when you eat meat very often, that will help bacteroides grow and not protein powders, right? Which is why also you'll see that vegetarians consuming a lot of protein powders have usually a flatulence problem,
I
right?
do. Yeah, I do experience.
ऑप्शन ही नहीं है, सबको ही होगा, और मतलब नहीं हो रहा है तो you are hiding it as good as that, right? So you will see that problem for people who are Prevotella dominant consuming a lot of protein powders, right? I have found my safe spot at whey isolates, right? And not the whey protein. So whey has two forms, the whole whey and the whey isolate.
It's yeah.
So the whole whey would have milk proteins into it, which my gut won't be able to digest because I'm not more animal source friendly gut, and which is why I'll have all the other problems like smelly flatulence, right?
Hmm.
I found it in whey isolate because it disregards all the milk components that come with the protein powder, and isolates are generally easier to digest for people who are more fiber friendly gut, right? So these, at least this has helped me. I'm also trying to understand this much better, but this is currently what has helped me, and this is my understanding after seeing about 3 to 4 thousand reports.
So I also consume whey isolate. It's the whole truth whey isolate
Right.
flavored this thing. But even after I'm done consuming it, so when I have it, I usually have it in a smoothie format, and I think that's why, if I've mixed a bunch of things, fruits, etc. I think all of that causes a little bit of bloating and flatulence, and I have to always walk around to sort of get that settled.
Yeah. I think that is there. I mean, so I had that issue as well when I was taking lots of protein powders. I don't want to name brands unnecessarily, but couple of the famous ones. Then I shifted to this new guys. I really like them, Protize. So Protize has really come out with a solution for this, that the protein is not very heavy. It feels like a juice, and the whey isolate is very easily digestible because of micronized particles, right?
Right.
So that has helped me. Even I had troubles with other brands, and I shifted to Protize, and now I'm feeling much better. I feel like I've just drank a juice and not a protein shake per se,
Yeah.
right? So
So,
yeah.
and now, yeah, with the whole the vitamin part of it, sorry, that is also something I was very, very, very interested in B12, because I think that definitely shifts how we take supplements, right? If everyone has this data, I'm sure a lot of the supplements that they may be consuming could be quite useless if they don't really know this. Or am I wrong about this?
No, see, how I would look at it is your gut microbiota can produce 4 vitamin Bs and vitamin Ks, right? Vitamin Ks we don't understand as much, so we don't report it. But 4 vitamin Bs it definitely produce, the ones that are there out here, right? Now, sometimes what happens is external tablets don't always work. Okay, could be absorption issue or could be anything. Our general logic is that if your gut is actually able to produce it, right, in enough quantities, maybe you don't have to go for a supplement at all, right? Maybe you can train the gut microbiota or eat foods which are more relevant to your vitamin production pathways so that it can release it, right? So that's the general logic we tend to go by, right? And yeah, sorry.
So where do the role of supplements come in? Say now, suppose again, just taking this report as an example, vitamin B7 and vitamin B12, I think I'm assuming I don't need to constantly keep supplementing for that. Now,
See, the idea here is that we are looking at genetic potential, not literal how much release is happening, right? So the genetic potential of your microbiota to produce B12 is very high, but maybe you are not giving it the right substrates for it to produce it at all,
okay.
right? So that's the difference, right? So for example, genetically I'm predisposed to diabetes, but if I make sure that my lifestyle is all right, then I can delay that genetic predisposition as well. Similarly, your genetic predisposition of your microbiota to produce B7 and B12 is very high, but it needs animal resources to produce that, which you're not going to get.
Yeah, I'm vegetarian, so that
Yeah,
there has to be a layer of because when I did my bloods, my B12 was on the lower side.
right.
So when I did my bloods, my B12 was on the lower side, around 300, 350, 370. And then I woke up to it, and I spoke to the bunch of experts that we have on our team, and they recommended that I take an intramuscular injection. I'm yet to get my levels done again, but I'm sure that they may have contributed in some way or the other.
Right.
So where does the interpretation of a report like this make for smarter supplementation strategy?
Surely. So I think for vegetarians, the option is only supplements for B12 particularly, right? Because it's the same thing, right? Let's say I'm a bacteria, okay? I'm really skillful at computational biology, right? But if you don't give me a server, what do I do of my skill set?
Yeah.
आपके बैक्टीरिया का स्किल सेट है B12 बनाने के लिए, उसको कुछ लग रहा है. The server for your bacteria is basically animal sources of food which it can consume, break down and produce B12 from, right? But you're not giving it that source because we are vegetarians by nature, and which is why your bacteria has the skill set but it's not able to perform because it doesn't have the resources. So for B12 you don't have an option, you have to go for a supplement, right? For me and you who are more vegetarians in nature, right? So that's the issue here, where we say that animal food, non-vegetarians don't have a B12 issue, right? That's the usual interpretation of how we have understood the subject. But that's also the B12 in a non-vegetarian is also produced by the microbiota using the animal sources, pretty much. So that's the issue here. Even the point is that despite your microbes having the skills, this part is useless for you. You still have to go for supplements because you're not going to consume meat, which your bacteria can produce and convert it to B12.
Got it. And on the dopamine serotonin part, where that is, uh,
Yeah, just
which page is that on? I'm sorry.
I'm coming.
Yeah, yeah. So yeah, this now again, what are some of your top level interpretations from this?
Hmm. So first of all, serodopa we have generally seen coming out low in Indian population, right? It's also because of the bias that we have tested more urban population than rural so far. So we don't exactly know what's happening on the rural side of things. But generally, serodopa we have seen low enough. We ask people not to worry about it much because the general understanding is that maybe gut-derived serodopa is low, but that doesn't mean the brain-derived is also low, right? So that's the first part we need to be very clear of. This might help you a little bit with gut motility and everything, right? So the usual symptoms that we'll see on an everyday basis could be something like bloating, or could be something like as soon as you eat, you want to evacuate, right? So that's the gut motility issues which people like us in urban areas usually have, right? It's not very prevalent or it will not affect us a lot, but sometimes every once a week or something will have these minor issues because of that. And that is because innate capability of somehow Indian guts to produce this is low, right? That could be one reason. The second reason, which we are still exploring, is that do we have the right references to understand if these are the only genes that are contributing to serodopa production in the Indian gut? So that's a very technical hypothesis that we are trying to solve as the whole scientific community of India, not we alone.
So I'm guessing you'll need a much more diverse and much more representative biobank to arrive at a stronger conclusion for that.
100%, 100%, yeah. And much more controlled cohorts as well, right? So our controlled cohorts comes with collaborations with academic institutions like Indian Institute of Science and AIMS, and collaboration with pharma companies, collaboration with hospitals, where we get a certain disease, lots of data about that, and then we can really start segregating that data generally into what's more relevant for you.
Got it. Got it. Yeah, so I think the report, I think I'm done picking your brain about the report now. So I had just one section that is now something that people are talking about a lot, which is the role played by antibiotics and what damage or support they give to gut bacteria and the prevalence of pathogens versus good bacteria that we clearly saw. So what is typically the correlation over say about sorry so what is typically the correlation over say about you know any major breakthroughs that we've seen that has shown that antibiotics have negatively or positively affect gut bacteria, or they don't have any effect?
Oh, antibiotics is the biggest blessing and curse for the gut microbiome scientists or someone who's having a gut related issue. Reason being, we have two types of antibiotics generally, a broad spectrum one and a narrow spectrum one. Broad spectrum means whatever microbes comes in its way, it will just kill. Narrow spectrum, it will select certain bacteria and kill them, right? Generally, what we use is a broad spectrum antibiotic. For example, azithromycin, right? Very common to take. Now, we are all aware about what recently happened with Operation Sindoor, precision strikes versus all-out blown-out attack, right? So that's the difference of antibiotics of broad and narrow spectrum. Usually, antibiotics is a bomb blast, right? It will go in your blood, it will create this big blast, whatever comes in its way, it kills, right? Which is why people who are on a repeated dosage of antibiotics will always complain of stomachache and diarrhea. Option ही नहीं है. There are no bacteria. Good bacteria die who actually digest your fibers and make SCFAs. When they die, fibers don't get digested. When fibers don't get digested, it's all going to come out as diarrhea, right? So that's the first issue antibiotics will cause. So it's very clear that antibiotics are bad for your gut microbiome, right? Even for your lung infection, you take an antibiotic by your mouth, okay? It will help you with lung infection, but it will kill all the microbes in your gut, right? You have a skin infection, you're given an antibiotic, it kills all the gut bacteria, helps you with your skin, right? So this is the issue with antibiotics. The bigger issue is that when you take repeated antibiotics, right? So your bacteria is going to get resistant to it. So even the pathogenic species one, right? If someone slaps you every day, at some point you're going to be like, your slap doesn't hurt me, right? So that's what's happening. So many Indians are taking more than 3 courses of antibiotics in a single year, which is crazy. It is unprecedented ever in the history of humankind. And you're destroying your microbiota. Your microbiota is acting against it, reserving antibiotic resistant genes, which means they can fight off this drug next time. That means next time you have to take even a higher dosage, and then your gut again gets resistant to it. So you have to take even a higher dosage. This keeps continuing, and it's going to cause a massive issue. So the next epidemic, as gut microbiome scientists says, is antibiotic epidemic, which is that you will not be able to cure anything with the existing antibiotics. You'll have to come up with something new. The research has already started. Recently, only someone developed an antibiotic which will not cause antibiotic resistance, right? So this is one of the bigger issues. So I wouldn't say antibiotics are bad, but sometimes waiting out helps better than antibiotics. What happens is minor gut discomfort. आज सुबह मेरे को डायरिया हो गया या तो आज सुबह मेरे को ब्लोटिंग हो गया, मैं सीधा जाके एंटीबायोटिक ले रहा हूँ. That's not right.
या गले में खराश हो गया एजिथ्रोम,
मैं एंटीबायोटिक ले रहा हूँ, राइट? कुछ भी हो रहा है मैं एंटीबायोटिक तो ले ही रहा हूँ 3 दिन. Now that is a bigger issue. Sometimes just wait it out, right? You have a throat infection, antibiotic will cure it in 3 days, increase your gut symptoms for next 15 days. Rather than wait for 7 days, the infection will go. Take vitamin C's or vitamin D's. The infection is going to go in 7 days and your gut will be preserved at least. That doesn't mean you take a lot of vitamin C because it also affects your gut, but still, it's a better way to manage it, right? So there are three techniques to deal with gut-related infections, right? If there's H. pylori or C. difficile, surely take an antibiotic. You don't have an option, right? But minor discomforts, minor infections, either you wait it out or you have other options which are coming up in research nowadays, or you wait out for that. So that's my general understanding of ABX and microbiota.
So we discussed a lot when we were reviewing my gut microbiome report about what general observations you see in Indians. Now, one of the and you said that you've not studied rural populations much, but there are certain adverse findings that you've commonly seen in urban Indians as well. Um, my belief, and I mean, obviously, it's not validated because I don't go to every sabzi mandi and every other place that is selling vegetables and fruits. But is that because we're such a big country in terms of population, and there's just that much land bank to feed all of us, the use of pesticides is very prevalent in our food. Given all the bacteria that you showed me, which pathogen do pesticides increase the balance of in your gut if you are having pesticide laden food?
No, I don't think so. There's a direct correlation like that, where
Okay.
having a pesticide will help a pathogen grow. It doesn't work like that. Also, we don't have a lot of studies in context to Indian population about this as well. But what I've learned from international studies that have happened in urban centers is that two main findings come across, right? So usually if your food is heavy in pesticides, your urine would have certain kind of metabolites, which can tell me if you have consumed pesticides or not, right? So when people studied that urine metabolites and their gut microbiome, they realized that people who have high presence of pesticide metabolites in the urine also has a low diversity of good bacteria and usually are more inflammatory in nature, which means all these LPS and cytokines and IL-6s are generally released. So there is definitely a correlation about having pesticide laden food and your gut health going bad, right? So that's first kind of correlation. The second kind of correlation, I completely lost track of my thought there. But yeah, yes, the second part, which is coming up very interestingly in the literature, which we don't show it in the reports and we are still discovering ourselves, is that urban population has also evolved to have more genes in microbiota, which are xenobiotic degraders. Xenobiotics means it's like extraterrestrial things for your microbes, which they're not used to since ancestrals, right? But new things come. Now bacteria also fight. To fight it off, they have xenobiotic degraders or certain molecules which help them counter these bad molecules that come to them, right? So there is definitely a more presence of genes of xenobiotic degradation in urban population. How they are converting biologically, we do not understand much, but I think that's an area of research which is really, really progressing well in microbiota.
And now the one thing again that's been a trend in the last, say, 3 to 4 years is probiotic supplements, right? In India, the Good Bug, Sova Health, many other companies who have come with probiotic powders, supplements, and they even claim to have reduced weight by 43% and clean gut microbiome, reduce fatigue and energy crashes, etc. What have you personally seen, following this closely, and where do you stand on if someone even needs these probiotic powders or no?
Uh, I have a very, very mixed opinion on this, right? You will never see Decode Age so far doing probiotics, right?
Yeah.
Despite us doing the microbiome test. Because innately, my scientific team's understanding is that the maximum modulation to microbiota can only be brought about through food or hygiene. We still believe that at the core of our hearts. Coming to probiotics itself, right? There's one or two things which I completely trust in the probiotic science, which is Bacteroides fragilis, right? I absolutely love that guy because it causes a cascade of metabolic reactions, which eventually releases GLP-1 hormone inside your gut, and that actually helps you out,
It
right?
slows down appetite, I'm assuming, right?
Yeah, and that's what helping with the weight part of it at least, right?
Yeah.
But obviously people have done the clinical trials, have shown the results, but I also feel that it's not working for everyone, because the biggest problem in probiotics, which you don't see people discussing, is colonization efficiency. Very technical term, okay, but it's as good as something like that. Your gastrointestinal tract is a pipe. Okay, it's open on one end, open on the other end through your anus, and बीच में pipe है. ठीक है? You put something in the pipe, right? It comes out.
Yeah.
If you want to make something stick within the pipe, you need to have the right mechanisms for that, right? Now we don't understand that mechanisms. Almost 7 out of 10 trials which has happened for probiotics showed that the probiotics that was consumed was never found in the gut after we took the medication. That means it just passed. ऊपर से नीचे निकल गया, right?
Okay.
And the benefits, जितने लोगों को benefits हुआ है उतने लोगों को side effect भी आया है.
हाँ.
ठीक है, ये कुछ ना कुछ. अब देखोगे काफी लोग बोलते हैं यार प्रोबायोटिक लेके मेरे को सडनली डायरिया शुरू हो गया. ठीक है,
Yeah.
बिकॉज शायद वो कुछ और चीज भी लेके जा रहा हो. We don't understand that. So this is the biggest problem in probiotic science, which is the colonization efficiency. We do not know if the species that we are consuming are sticking to the gut or not. Secondly, I'll bring back to that topic. The second point is that we don't know if the US wala Prevotella copri and India wala Prevotella copri are the same. हम क्या कर रहे हैं, हम यूएस वाला प्रिवोटेला कॉपरी बना के खा रहे हैं, राइट? But we don't know if it's actually helping or harming the gut on short or long term. So that's the second problem we see. Third is almost all good benefits that have been reported with probiotics in the world are for strains and not for species, right? That means that, let's say Karan Sancheti has 5 twins, right? But 5 twins will act differently, although you all look the same and you all are from the same family, right? So that's the difference. And we do not have enough understanding about these strains in India. Secondly, it's very difficult to stabilize a strain which is made in a lab, to capsule it, to transport it over 35 to 40 degrees of temperatures, it coming to your place and then you consuming it, surviving through the stomach acid, surviving through the small intestine, reaching the large intestine and colonizing there. ये किसी को पता नहीं stability है या नहीं ये पूरे process में, right? So I would say there's still a lot of good effects are placebo in nature. Okay.
Yeah.
And if there are certain good effects for that, it's not consistently or statistically observed through populations at multiple levels. That's my overall understanding of
So it
probiotics.
remains to be seen if there are any one-size-fits-all conclusions that might come out of this, right? But right now there aren't any, I'm assuming.
There aren't even personalized probiotics.
Okay.
It doesn't work, right? Just because you don't have a certain species in your gut, taking it doesn't mean that you will have that in your gut. You and I eat curd every day. My gut is not able to harbor lactobacillus, right? Which is a part of curd. I eat curd every day, still lactobacillus is not there. That means that something about my gut is not allowing those guys to live. And we do not understand what that something is. So lots of loopholes.
So before the gut microbiome existed and, people were still getting blood tests done, what are the blood markers that we need to look out for that can be, secondary or tertiary indicators of good or bad gut health, assuming we could test across markers?
CRPs are your biggest this thing affected, which is the inflammatory panel, C-reactive proteins, right? Because if your gut is not in the right homeostasis, it's gonna continuously create this low-grade inflammation, which is going to manifest in your blood reports in CRPs or HSCRPs, right? If you don't have an infection and you're still HSCRP is very high, your gut is definitely going through something which you need to look out for. So that's the first place you'll get. Hepatic profiles or the liver profiles also get affected because there's a continuous crosstalk between gut, pancreas, liver, and the insulin or glucose pathways, right? So that's also working in a certain direction. And third is the glucose profile, right? If your diet is generally healthy and if you're still not able to bring glucose or HbA1c in control, that means something is going wrong in your gut, which is not allowing the glucagon pathway to function properly as we would like it to.
Got it. And One last question before we conclude. What is that one piece of advice which is most universal that you have seen working for clients that you guys have dealt with across studies, across your experience as a student, researcher, and now even in the business of gut health? What is that one piece of universal advice that you think is almost always likely to work for everyone, or maybe three, four, whatever it is?
Sleep and dietary habits, and that's like the two main things that will help your gut, right? Sleep, because somehow the circadian rhythm and gut microbiome are connected. We are not really sure how, because we don't have evidence, but these two guys are definitely connected, right? So sleep really helps. If you're sleeping fine, your gut is going to be fine. That's the one bottommost. The second is dietary habits, and not getting carried away with all the things that you hear about a certain kind of dietary habit, right? मेडिटेरेनियन डाइट का सुन लिया तो अब मैं मेडिटेरेनियन डाइट खाऊंगा, ठीक है. ओकिनावा का सुन लिया तो मैं ओकिनावा काइंड ऑफ फूड खाऊंगा, ठीक है. देन वी हैव लो फॉडमैप, which is like the king in gut microbiome science. Low FODMAP is basically less fibers in your gut. Low FODMAP only helps when your gut is already bad and you want to starve off the bad bacteria so that they can die. But then you have to replete it with fibers and probiotics food, right? If you're not doing that, you're done and dusted. Your low FODMAP forever is going to uh, be more harmful for you than a good diet
Yeah.
overall. The reason we still ask people who are onto their health journey to do microbiome is to make sure to understand what is good for you in terms of food items. Okay, how can you personalize your diets? We will never recommend you probiotics. We will never recommend you supplements, right? Because there's nothing that can control that. The only thing that we know that can control that is diet and personalization to a certain extent.
So
Things like, why is spinach healthy for me and unhealthy for you, right? Questions like this you need to ask. And that's what matters, I feel, the most.
got it. Awesome. Thank you so much, Darshit. I think this was my most educational podcast. I think I've learned so much more about gut health, gut mind axis, how the gut microbiome actually plays a huge role in our bodies. And I think and I'm very, very thankful that you took out the time to do this.
No,
Thank you so much.
no, of course, Karan. I think the questions are very well designed, usually. And I think the way they were designed also led me to answering certain very basic facts with the science behind it. So, I mean, commendable. I absolutely enjoyed having this conversation as well.
Thank you so much. Thank you so much, Darshit. Pleasure was
Cheers.
mine.
Cheers, Karan. Thank you. Thank you so much.